Did they both load? No. Slide. You were on. Inhibitors. I'm so glad I remembered my jacket today. All right, so y'all ready? Please make sure those masks are up for mouths and noses. Everybody masked up. Thank you. All right, so how fast is milk? It's pasteurized before you know it.
Oh, I thought you were going to say it's not very fast because of lactose.
That's a really good one, isn't it?
It
It was great. I had to explain it to Charles. It was kind of a disappointment at home because then we had to spend the morning teaching what is pasteurization. But it was still fun. Okay, so before we start today, I do have to make one announcement. I made a decision and I moved our exam back a day, our next exam.
You actually did it.
I actually did it. I never do this, y'all. I like never.
You actually did it.
I did.
You're a math. Oh my God.
So, so I want everybody to realize this ahead of time. I am not moving it from this date. Okay. So it was on the Wednesday and I moved it to the Thursday. So this is, that's what I meant. It was on Wednesday. I moved it to Friday. Sorry. So this is next Friday, not this Friday, but next Friday. It was going to be the exam on Wednesday. Now it's the exam on Friday. We good?
Yes.
Homework for chapter 7 is due tomorrow night because we're going to finish it. Y'all listen? Yeah. And then I pushed back chapter 8 homework because I'm anticipating that it'll take us through Wednesday to finish chapter 8. Okay. All right. Moving on. What am I? I gotta be on this. Okay. So we were talking about enzyme inhibition, right? And we said there were two general categories. There was reversible inhibition and there was irreversible inhibition, right? Irreversible talks about covalently linking that inhibitor to the enzyme and it basically kills the enzyme, whereas reversible, we can decrease it by diluting the enzyme, by diluting the inhibitor, and then our enzyme will become more active. Okay. So here's an example. Here's an example of malonate. This is a reversible inhibitor of an enzyme called succinate dehydrogenase. And when we get to glycolysis and citrate cycle and electron transport chain, you are going to see that all of these examples that I've been using throughout the entire semester are all enzymes that are in those pathways. And so that'll be like the end of this semester that we'll talk about that. So what do you notice? Here's our substrate. This is succinate. Here's our enzyme, succinate dehydrogenase, and it makes fumarate. So here's our inhibitor, malonate. Can you tell me, are there some similarities between the substrate and the inhibitor? Absolutely, absolutely. A lot of times for reversible inhibitors, our inhibitor is going to look like the substrate. If the inhibitor looks like the substrate, more than likely, where is that inhibitor going to bind to our enzyme? At the active site. Exactly, at the active site. So basically, there's one active site and only one thing can bind. If succinate is there, it's going to bind succinate, it's going to make fumarate. If malonate is there, it's going to bind malonate, and because it's an inhibitor, you are not going to make the product, right? So it's kind of an either or sort of deal. But that's not the only possibility on how inhibitors work. There are actually three major classes of inhibitors. I move this back up. Participants can now see your screen. I hope they were able to see it the whole time. I hate when that happens. Okay, so we have three different kinds of inhibitors. We have competitive inhibition, we have uncompetitive inhibition, and we have mixed inhibition. So what we're going to do is we're going to generally talk about all three, and then we're going to give some examples of them and look at how they change the kinetics of chemical reactions, right? So kind of draw yourself a line every day. Draw yourself a line so that you can kind of keep your notes separate for each of the different inhibitors, right? So if you have something that is a competitive inhibitor, a lot of times a classic competitive inhibitor is going to bind at the active site. And that's exactly what we just saw on the previous slide. So once it binds, once the inhibitor, which is the little pink thing here, binds at the active site. So where's the enzyme's active site? Right here. So either you can have the substrate bind to the active site, or you can have the inhibitor bind to the active site. But you can't have both, right? So what's going to end up happening here is that either it'll bind at the active site, or in non-classical inhibitors, competitive inhibitors, it can bind to part of the active site. So if you block a portion of the active site, then your substrate still can't fit, right? So the concept is still the same, but it might not take up the entire active site. Or there's a third possibility. That inhibitor can bind away at a distant, we'll say a distant binding site. How does that work? If I have an inhibitor that's going to bind at a site that's far from my active site, how in the world is that going to inhibit substrate binding? Exactly. We have a distant binding site. So there are three. There's the classic, there's blocking part of it, and then there's a distant binding site, which then alters conformation of the active site. Right? That good for everybody? Three different possibilities. Yes, ma'am.
And that would still be competitive.
That would all be competitive. See if I can do that. Because basically what you're saying is that this enzyme can only bind one of two things. It can only bind either the substrate or the inhibitor. And once it's bound to one, it can't bind to the other. No. Classic, like the classic, it doesn't really matter. You don't really have to know that it's named classic, but the typical examples that you're going to see is where the inhibitor binds perfectly inside the active site.
So it's competitive in the sense that they're fighting for the enzyme, not the active.
Correct. In classic, they're fighting for the active site. In competitive inhibition in general, they're competing for the enzyme. Yes, that's exactly right. Yes, ma'am. Yep. Okay. All right, so now we're going to go to our other types. I'm going to change, I'll try to change color. Let's see. Oh, usually there's a way to change. There we go. Okay, so now uncompetitive inhibition. In uncompetitive inhibition, you have the enzyme, you have the substrate. The enzyme binds the substrate and normally will make it a product, right? In uncompetitive, once that enzyme substrate complex is made, now the inhibitor can bind to the enzyme and inhibit product formation. So are they competing with one another? No, they are both the substrate and the inhibitor are bound at the same time. So you have enzyme substrate inhibitor complexes. You have no, here you have no enzyme inhibitor complexes. Do you see that? See the difference? Here I have enzyme inhibitor or I have enzyme substrate. So here you have enzyme inhibitor or enzyme substrate. Here the only thing that you can have is what? You have only enzyme substrate inhibitor complexes. So basically what happens is when this inhibitor binds the enzyme substrate complex, it's actually going to induce a conformational change in that active site. Now that active site cannot do its catalytic activity. Even though the substrate's bound, the catalytic activity is inhibited and you cannot make product. Yeah? Okay. So then you can have, so before we go on, in uncompetitive inhibition, you're actually changing basically the catalytic activity of the enzyme, right? Are you changing the catalytic activity in competitive? No, basically, what are you doing in competitive? You're basically decreasing the concentration of enzyme. I mean, I'm going to put that in quotations because you aren't really changing the concentration. You're changing the concentration of enzyme that is available, right? But here, what are you doing? You're changing catalytic activity. Why do I have that blue line? Of the activity of the enzyme. Right? Yes. So in competitive, if I have the enzyme binding, the inhibitor binding here, let's just say, right? Then the substrate would not be able to bind. So you could only have enzyme inhibitor or enzyme substrate. Here, the inhibitor binds and the substrate binds. So you have enzyme substrate inhibitor complexes. And so when the inhibitor is bound, the enzyme can't do its job, but the substrate can still bind. Yeah. Okay, so let's do the last one. The last one is mixed inhibition. So if I do the same sort of setup, normally I have my enzyme that binds the substrate. I have my enzyme substrate complex, I make product. Now in mixed inhibition, two different things could happen. My inhibitor could bind to my enzyme, and when it binds to the enzyme, it changes the conformation. Or I can have enzyme that's already bound to substrate that then gets bound to inhibitor, which then changes the conformation of the active site. So you have basically both competitive and uncompetitive happening in mixed inhibition. That's why they call it mixed, because it's both. So a lot of times this one, I'll put it here, a lot of times this one, that the inhibitor binds at a distant site, binds at distant site. right? Because it would be very, very difficult to have something bind within the pocket and still have mixed inhibition. So a lot of times this is a distant. So you can say binds both enzyme and enzyme substrate. So if you bind the enzyme, you get enzyme inhibitor. If you bind the enzyme substrate, you get enzyme substrate inhibitor complexes. So you see both of these in mixed. In here, you're only going to see in competitive, you're only going to see enzyme inhibitor or enzyme substrate. And here, you're only going to see what? In the middle one. Only enzyme substrate inhibitor complex. Right? So if you look at the products that you have, you can determine what kind of inhibition is going on. Yeah? Yes?
So even without the substrate, it's still inhibited.
It still binds. Yes. It still binds to it. Just like here in competitive inhibition. If I had no substrate present, I would still have inhibitor binding to enzyme, right? In uncompetitive inhibition, that doesn't happen. In order for the inhibitor to bind, you must have substrate present. But in mixed inhibition, you can either have no substrate or substrate present. Either way will work. That's why they call it mixed. Okay. All right, so let's talk about each one of these in a little bit more detail, right? And a molecule that inhibits substrate binding at the active site. This is a completely true statement. It inhibits substrate binding at the active site. Does this mean that the inhibitor binds to the active site? No, right? It could bind and physically block the active site, or it could bind at a distant site that induces a conformational change that therefore results in a loss of substrate binding. Either way would be okay, right? So it can bind to the active site or obstruct the active site from binding, right? So how can you overcome this inhibitory effect of a competitive inhibitor? All you have to do is increase your concentration of substrate. If I increase my concentration of substrate, the enzyme is more likely to interact with substrate than it is inhibitor just due to concentration. So you can basically dilute out the inhibitor with substrate, right? Because if you have 5 molecules of each, substrate and inhibitor, and 5 molecules of enzyme, you have a 50-50 chance of getting either inhibitor or substrate. But if I change that and I say I have 9 molecules of substrate and only 1 of inhibitor, I'm much more likely to bind to the substrate. So then what's going to happen to your Vmax of your reaction? Whether you have inhibitor or not, Vmax is not changed. You're not going to change the maximum velocity of the reaction, but you do change the apparent Km value. So what is Km? Binding affinity, basically, right? So basically what you're doing is when you have an inhibitor present, you're decreasing the concentration of enzyme, or another way to say that is you are increasing the concentration of substrate that you need to reach a maximum velocity. Either way is true, and you don't have to know all the math. I'm really not worried about you knowing the math. I'm worried about you being able to analyze graphs. I'm telling you, you need to be able to look at one of these graphs and tell me what is going on. Hint, 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 hint. Y'all know what I'm saying? Yeah, okay. So, well, for all of the inhibitors, and we're going to talk about all of them. So if I look at my same thing, I'm graphing initial substrate concentration versus velocity of my reaction. So if I look at something that has no inhibitor, right, I get a particular Vmax. And I can go to 1/2 Vmax and I can come down and I can get Km, right? We did that last time, yes? Okay, So now if I add a competitive inhibitor, when I do that, your Vmax, you're eventually, as I increase more substrate, eventually I will get to my Vmax. But if I take 1/2 of that Vmax and I come across to the green line, whoa, what happened? My Km increased, which means I have a decrease in binding affinity. So it takes more substrate to reach a particular velocity because it is competing with the substrate is competing with the inhibitor. Yeah. So if I add more inhibitor, I'm still going to eventually get to Vmax. So if I take my 1/2 Vmax and I come across to the red line, what happened to my Km? It went up, right? Now, this can sometimes be hard to analyze. If we linearize it and look at it on a line weaver Burt plot, remember that this axis is 1 over the velocity, and it ends up being 1 over Vmax. That's our intercept. So what do you see? All of these intersect at the same place. So was Vmax changed? No. But look at Km. Now, do you see this Km? Do you see the APP? That's the apparent Km. So if I add an inhibitor, does my binding affinity actually change for my enzyme and substrate? No, it doesn't. You're just soaking up those enzymes, right? So it doesn't actually change the Km, but when you measure it, the Km is different. So we call that the Km apparent, because apparently you're changing the Km, right? So what do you notice about the Km? What happens to the Km? It's the negative inverse. So what happens to Km? Look here, what happened to Km? It increased, Km increased, Vmax stayed the same. So you have to be able to look at one of these graphs and tell me what kind of inhibition is happening. Okay, so let's look at a few more. Oh, this is an example. Quantivir is an example of a classical competitive inhibitor, right? And this is one that actually inhibits the protease activity of the HIV virus. And so this is one of the drugs that people who have HIV take on a daily basis to keep the virus in check, to keep the viral loads from being high and that kind of stuff. And so if you look at the protease inhibitor is going to cleave at these site between phenylalanine and proline. And so the inhibitor is actually going to mimic the natural substrate. So here's our inhibitor, here's our natural substrate. Looks gosh darn pretty similar, right? And so that's all it does. It just blocks the binding site. And so that's your typical classical competitive inhibitor. All right. So let's go on to uncompetitive inhibition. So in uncompetitive inhibition, you have to go back and remember what that means. That we're going to bind to our enzyme substrate complex, right? Are you going to have any enzyme inhibitor? Are you going to be able to form enzyme inhibitor? No, absolutely not for uncompetitive. For uncompetitive, you're going to have the enzyme that's going to bind to the substrate. You get enzyme substrate. Then the inhibitor is going to bind to that, and you're going to get enzyme substrate inhibitor complexes. So if we look at that, can I dilute out the inhibitor by adding more substrate? Would that work in the same way that it works in a competitive inhibitor? No, it wouldn't, right? It doesn't make a difference how much substrate I have. What makes a difference is how much inhibitor I have, right? Because the inhibitor is going to bind to the complex. It's not actually competing for the site. So if you look at the Lineweaver-Burke plot, what you're going to see is that you actually have a change in Vmax, right? Because you have these complexes that are locked. They can't do anything. So you're actually going to change your Vmax and you're going to change your Km. So you see both of them change in uncompetitive inhibition. Yeah. So there's so you could say even with, right, high substrate concentration, because that's what we did in the last one, enzyme is unproductive, right? It doesn't make product. And that's what makes Vmax lower. Yeah. So, okay, so this is similar to what we saw in competitive, right? Because are you actually changing the binding affinity of the enzyme to the substrate? No, right? The enzyme can bind substrate, no problem, and then the inhibitor binds. So you're not actually changing the Km. But when that inhibitor is bound to the enzyme substrate complex, now you have a non-productive enzyme, right? So because you are changing, you are taking out enzymes, you are sequestering them, right? You're going to change the apparent Km because you'll have substrate there, but the enzyme's not producing anything, right? So that's going to change your apparent Km. It doesn't change your real Km. It changes your apparent Km.
So real Km never changes.
Real Km never changes. It's always Km apparent. Yep. Mm-hmm.
With uncompetitive inhibition, it looks like that the Km parent is a wider range.
It depends. It
Or
depends.
is that just how the graph is drawn? Is that not?
It depends on the exact inhibitor.
Okay.
It depends.
So that's just a.
That's it. It's easy to visualize. Yeah.
I didn't know if that was like.
No.
Because uncompetitive was more effective. Or...
No. No. Yes.
And the real Km never changes with any of the inhibitions or just this one?
It doesn't change. It doesn't change. It just doesn't. Each enzyme has a particular Km for a substrate, and you're not going to change that Km. But when you start binding inhibitors, you change how the enzyme acts, which is the apparent Km. That makes sense. Okay, so then let's look at mixed inhibition, right? So whenever you talk about mixed inhibition, remember that in mixed inhibition, your inhibitor cannot bind to the active site because then you would be a different form of inhibition, right? So it's called mixed because you can bind the enzyme, you can bind the inhibitor to both the enzyme and the enzyme substrate complex. So this gets pretty complicated. We know that we're going to lower Vmax, but we don't know what's going to happen with Km. So if we look at these graphs, there's actually 3 different graphs. There's 3 different things that could be going on, right? You're always going to change Vmax. So look at all 3 graphs. What happens to all 3 graphs to our Vmax? What's happening to Vmax? Our Vmax is decreasing, right? Okay, now what else is happening to Km? Well, here in some places, Km doesn't change, right? So what other graph did Km not change? Go back. Ooh, I went too far. Okay, so here, did Km change? Yes, but Vmax did not, okay? So if we go to uncompetitive, Vmax changes and Km changes. So when we look at mixed inhibition, this is the only time where apparent Km is not changing. So apparent Km can stay the same It can get smaller, it can get larger. It depends on the inhibitor, but you're always going to see a change in your Vmax. Yes? Okay. All right. So remember how we talked about things that were covalently inhibitors that covalently interacted with our enzymes, right? When they have a covalent interaction, what happens? Oh, what did I say? You said for competitive inhibition. So if we go to competitive inhibition, our Km does not stay the same. Our Km changes, but our Vmax stays the same. So here Vmax is the same, Km changes. All right. So if we look here, this is an example of an irreversible inhibitor. So when we look at those enzyme mechanisms, right, and we were looking at covalent catalysis and things like that, that's exactly what happens in irreversible inhibitors. So remember how we talked about proteases, things that had the triad, remember that? Okay, it's the same sort of deal, where we have these side chain residues that are inside the active site that interact with the inhibitor. And when they interact with the inhibitor, they create new covalent bonds between the substrate and the enzyme. And then that enzyme is done. Like it's not gonna, there's no regeneration process just because of the way the inhibitor is designed. And so this is a huge pharmacological field, right? If you can make enzymes that make permanent covalent bonds with inhibitors, you can pick and choose what you want to inhibit and what you don't. That has huge therapeutic potential, right? It's not a temporary fix, it's a permanent fix, right? The cell can't dilute it out. So there are lots of clinical targets for this, which is really, really cool. Okay, so when we talked about this section, when we talked in this section was enzyme regulation, right? So we talked all about how inhibitors can regulate enzymes, but you can have enzymes that are modified. And this is the suicide. That's what I was talking about. I'm sorry, I forgot I had that slide. But this is irreversible inhibitors. That's what we talked about, all of that stuff. Okay. So the other thing that you can do is you can modify the enzyme. You can add a covalent group to the enzyme to turn it off and on, or you can use a binding partner that induces an allosteric change in the enzyme. And the one that I'm going to show you as an example is an enzyme that's required to make CTP, right? So we're talking about our nucleotides. So this enzyme works very, very early in the enzymatic scheme of things. And so a lot of times when you think about how do I want to regulate an enzyme, I want to have feedback inhibition, right? So if I have this pathway and at the end of the pathway, whatever my final product is, I want that to be able to come back and inhibit one of my very early stages. Otherwise, I have this work that the cell is doing that's wasteful, right? I'm using up resources that I don't really need because I already have product. So a lot of times what you'll see is the end product comes back and inhibits some of the early stages. And the other thing that you're going to see over and over and over again is that when you're looking at a biochemical pathway, all of these things require energy, right? So if you haven't eaten for days, do you really want to be making new stuff? No, you kind of want to just be surviving, right? So a lot of times the energy charge of the cell is going to control a pathway or not. So if we ate recently, we're going to have lots of what? What's our energy currency? ATP. We're going to have lots of ATP. So we need to have our end product inhibit. And at the same time, we're going to have the energy charge of the cell regulate. So if we have lots of ATP, that ATP can be a positive regulator. So that's what we see here. CTP is an end product. This is an end product of the pathway. And this ATP is our energy charge. So the end product is going to be the inhibitor. And this is going to promote activity of the enzyme. So if you have just CTP present, the enzyme induces a conformational change by binding to some kind of regulatory subunit to make that enzyme inactive. But on the other hand, if you start running out of CTP, you're not going to have any to bind here. And let's say you have lots of ATP. That means you've eaten recently. Well, then go ahead, make some more DNA, make some more cells, right? So then the enzyme becomes active and it can do its job, which you don't have to memorize the pathway. You just have to understand how enzymes can be regulated allosterically, right? There's a really great animation of how this works. Like really, it's awesome. You should go and watch it. I put that in there every time. Y'all notice that? Like every time I put that in there. It is. I love their animations. I love this book's animations. They're awesome. All right. So when we talk about allosteric regulation, right? We want that feedback inhibition. We want the end product to function as the inhibitor of, it's not always the first, but I would say an early, E-A-R-L-Y, an early enzyme. Okay. So the example is the one that I just showed you in the previous slide, is ATC case. But you don't want to waste energy making metabolic intermediates that you don't need. It's a drain on the cell's resources, and the cell doesn't like that very much, right? So this is just the same pathway that you saw on the previous page, but just structurally, right? Here's our end product. Our end product is CTP, and the CTP will come back as soon as you make it after all these pathway steps and inhibit one of the initial enzymes. It's not the first step, but it's one of the very early steps. Okay, so you don't have to memorize this particular pathway, but you might have to analyze a graph that looks at allosteric regulation of an enzyme, right? So if we look at our control, that's the green line, right? This is no inhibition. This is no promotion of activity, none of that. So if we add lots of CTP, what should happen to our reaction rate of our enzyme? Up or down? If we add CTP, which is the product of the entire pathway, what should happen? It should go down, right? Our velocity should decrease. That makes sense, yes. But on the other hand, if we put something that stimulates the enzyme, that promotes the enzyme activity, like ATP, what we should see is an increase in the velocity. And this is very typical in that you're going to see a greater effect with an inhibitor than you do with stimulatory agents. Okay. So the other way that you can allosterically control enzyme regulation is through covalent modification. What have we talked about so far? The only one we've really talked about so far. So it's an on-off switch. Phosphorylation. We talked about phosphorylation over and over again. So that is one thing that can be added or removed from an enzyme. But you can also have a methyl group, an acetyl group, an adenyl group, a uridyl group. Like there's a bunch of them, right? But the most common is when we phosphorylate serine, threonine, and tyrosine residues. What do serine, threonine, and tyrosine residues have? So you're adding that phosphate group to the OH, right? That's why those residues are really, really important. So kinases add the phosphoryl group, phosphatases remove the phosphate group, so that it can be that on, off, right? It's not permanent. You can pull the phosphate group off. So here's an example, glycogen phosphorylase. So this one is, if you haven't eaten in a while, your body is going to try and feed your brain glucose. So you have stores of glycogen in your liver that you can metabolize and send. You can break down the glycogen and then send out individual glucose monomers out to the bloodstream so your brain can pick it up. That's your brain's preferred food source. So depending on whether or not you are phosphorylated tells you the activity of your protein. Well, what controls whether or not you're phosphorylated? And it is hormones, right? So glucagon is something that your body releases when you haven't eaten in a while, right? Your blood sugar drops, you start releasing glucagon so that your blood sugar will go back up, right? So if you add glucagon, the other one is epinephrine. So like if you're in fight or flight, a bear comes in the back door, right? You're activating your fight or flight response. And so to give you a little extra energy, to run a little faster, to see the bear a little better, you know, all that kind of stuff, you get a little extra sugar in your bloodstream.
I really hope there's no bear in the back door.
I always use that analogy. I don't know why, but I always use that analogy. I think because it's so never going to happen, I don't feel bad using it. So either way, you're going to activate phosphorylase kinase. The kinase is going to add a phosphate group to serine. Now, on the other hand, if you just had lunch, your body's going to produce what? Insulin, right? Insulin is going to activate protein phosphatase 1. Protein phosphatase 1 cleaves this phosphate group off and you have the inactive state. So this is how hormones can regulate enzyme activity to then have an effect on cellular function, right? Isn't that cool? That's awesome. Okay, the last way that you can regulate enzyme activity is through proteolysis. So there are some enzymes that when they are made, right, they're proteins. When the proteins are made, they are complete and they are inactive. And in order to become active, they have to be chopped. They have to be cut. So they become active upon cleavage. So here's an example, pepsinogen. They call it pepsinogen because this is the inactive precursor. So that's where the gin comes from, right? It's not an enzyme yet. It's a zymogen, it's a precursor. And then when pepsin is active, you actually have cleaved off this orange portion. Like this whole space filling, all of this is one polypeptide.
So it's kind of like a kinase with a built-in inhibitor.
Correct. Part of the molecule itself is an inhibitor. And you have to chop that little piece off. And once you chop that little piece off, oh my gosh, now the binding site is exposed and our substrate can bind inside of that cleft. A lot of times it's a pH change that will allow that cleavage to happen. So chymotrypsinogen is another one. What do you think the final product is going to be? Chymotrypsin, right? So chymotrypsinogen starts out as one entire peptide. If you cut it once, you're going to get trypsin, right? Depends on where you cut it, what you're going to eventually get. And then by the time you get to chymotrypsin, you've cut and you have 3 portions of the initial polypeptide that fold together to make a functional chymotrypsin enzyme. You don't have to remember all the different cleavage sites. You just have to know that some enzymes can be regulated by cleavage and that they're called zymogens. They get cleaved, they turn into functional enzymes. Okay. Questions on that? Good. Okay, changing gears. Changing gears. Chapter 8. I feel like it was good we spent a little extra time on that stuff, because that's kind of hard. So for those of you who are biology people who are like, God, that was a lot of chemistry, you're gonna love the next chapter. For those chemistry people who were like, yes, I was having so much fun, I'm sorry. It's gonna be a lot of biology. It's cell signaling systems. Chapter 8 is cell signaling systems. This should overlap pretty nicely with what class? Physiology. Yeah, see, it's gonna be so awesome. So awesome. All right. So,
You're kind of assuming that we like physiology.
you don't like physiology? Oh God, I love physiology. All right. So these first 3 sections is where the majority of the content's going to come from. We're not really going to talk about tumor necrosis factor receptor signaling. We're kind of going to like cover it like in one slide. And then we'll talk some more about nuclear receptor, but the majority of the bulk of the material comes from these 3. All right, so when you think about cell signaling, now you have to kind of go back to remember what we talked about before, right? We have a ligand. The ligand comes in and binds to what? The receptor. This binding induces what? A conformational change in what portion of the receptor? The intracellular portion of the receptor. That is the transition of the signal from the exterior to the interior of the cell. So that conformational change will propagate in a cascade of events. So when we look, here's our green thing is our receptor, right? That receptor usually has things very closely associated with it at the membrane. And so it's going to change its binding partners. It's going to be able to phosphorylate something. It's going to be able to do something to its friends that are really close to the membrane. We call those upstream signaling proteins. And there's usually a couple of them, right? And those upstream signaling proteins, when they change their binding partners, when they become phosphorylated, when they do whatever it is, they are going to activate what are called second messengers. Second messengers are things like cyclic AMP. There's a bunch of different ones, and we'll talk about those. But they're molecules that basically transition the signal from being localized to the plasma membrane to going someplace else in the cell. So those secondary messengers then activate downstream signaling proteins. These downstream signaling proteins are the proteins that actually modify the target proteins. So right at the end of this whole massive cascade, what do I want? I want protein phosphatase 1 to be active, right? That's my end game. Well, the downstream signaling proteins are the ones that really change the cellular activity, right? And so there are lots of different targets. It's not just one. It's not just metabolic enzymes, which is kind of what we've been talking about so far. But sometimes it's a transcription factor, right? We want to change expression levels of a particular gene. Maybe it's a cytoskeletal protein, right? Maybe it's a white blood cell. And instead of floating around in the bloodstream, I want it to enter into cellular tissue. So you actually have to change the skeleton on the inside of the cell to force it in between the epithelial cells of the blood vessel, right? Or maybe it's a transport protein. I need to ship something in or I need to ship something out. Either way. But I do like this system because they were actually pretty nice in how they name things, especially towards the top, right? Whatever your first messenger is, typically your receptor protein name matches that. So if you have insulin, guess what your receptor name is? The insulin receptor, right? You have estrogen, it's the estrogen receptor. You know, they actually did you a solid and made it a little less confusing. Right, they got you back, they got you back. Okay, so everything that we talked about just now is all forms of what? That we've talked about before. In the very beginning, signal transduction, right? Taking a signal from the exterior of the cell and turning that into a cellular functional change. So this mechanism that we're going to talk about is going to facilitate that change. And it changes your information into chemical signals, right? That's what you're looking at inside of the cell is chemical signals, right? You have increase in cyclic AMP and this happens, and you phosphorylate that and that happens, right? All of those are chemical signals. And so usually at the very end of this massively long process, you end up with a covalent modification of a target protein. And typically, most often, it's going to be phosphorylation or dephosphorylation, right? So all those things that we just talked about, metabolic flux, transport, cell motility, gene expression, we just discussed, those are all endpoints, right? This is what the target protein controls. So the target proteins control these things. Okay. So let's kind of start at the receptor. So what happens when you activate a receptor? You can have different things occur. Sometimes when you activate a receptor, you get covalent modifications of the receptor on itself. So there's not another protein that needs to interact for that phosphorylation to happen. Some receptors have inherent kinase activity and can phosphorylate themselves. It's kind of cool. You can change the conformation such that you alter binding interactions, right? So you could now associate with something that you couldn't associate with before, or you lose an interaction. Either way, you can gain an interaction or lose an interaction. And remember, some receptors are not on the membrane. Where are some receptors? In the nucleus. And what are they? They are transcription factors. So sometimes binding of the ligand, right, the extracellular, they'll just go right through the membrane, right through the plasma membrane, right through the nuclear membrane, and they'll actually bind to those transcription factors, which then do the job right away. So there's not a whole lot of steps when you have a transcriptional control, right? If it binds to the transcription factor, it either activates the transcription factor or inhibits the transcription factor. So you either start making more of the gene or you make less. So it's a whole lot fewer steps if you're talking about a nuclear receptor versus a cytoplasmic, a membrane receptor. All right, so we call these biochemical reactions the cell signaling pathway, right? You learned this in physiology, right? Heard this before, maybe. So you're always going to initiate by your ligand, right? Your ligand is going to activate your receptor. And then at the end, you have to have some kind of a measurable cellular response, right? But there's a lot of stuff in between there. And we're going to learn that the stuff in between there are dependent upon the type of receptor it is. And we'll look at lots of examples. So you can have things that are first messengers. First messengers are always going to be ligands that bind on the exterior portion of the cell to your receptor. So give me an example that we've talked about today that is a first messenger. What? Epinephrine, right? Insulin, glucagon. Sometimes it can be other things. Sometimes it can be ions, Ca2+, right? It can be a variety of different things. So this first messengers, it's a variety, right? It's all kinds of different things that could be first messengers. Second messengers are inside the cell. Second messengers translate that information from basically the collection of proteins that are at the plasma membrane to whatever the target is inside the cell, right? So a lot of times secondary messengers are like cyclic AMP. That's supposed to be a little c, not a big C. Cyclic AMP. AMP, as I'm, oh Lord. What's that? Yeah, makes you want to go see a movie, right? I'm ready to see Dune. So here are our secondary messengers, right? So our secondary messengers link those upstream signaling proteins to the downstream signaling proteins. So go back and add that. So these are our link from upstream signaling. Ooh, can I extend the page? Upstream signaling, GNA, signaling to downstream signaling. All right. Okay. So we have that. Now, when we talk about, let's talk in detail about some examples of things that can be first messengers. Hormones are an example, right? These are typically usually produced in one place, travel through the bloodstream to their target destinations, right? So there are a couple of different ways that you can get hormones to particular places. And so it depends on where the cell that's making it is, and then where is the target cell. So you typically have three. You have endocrine, paracrine, and autocrine. Talk about autocrine. What does auto mean? Self. So this is a cell that makes the hormone and is the target, right? That's autocrine. Okay. How about what's the difference between paracrine and endocrine? Paracrine is cells that are close or far away? Close. And then endocrine are cells that are far, and we're going to say from target. Correct. Correct. The time. I see everybody packing up. Yes, it's time. It is time. So glad I moved the exam. All right, y'all have a good rest of your Wednesday. We'll pick up there next time. Maybe on Zoom need anything? All right, but I'm going to go ahead and end.